Hello again and welcome back. In this video, we will focus on what types of information you should be putting in your approach section of your proposal. First, what I want you to do is think about what is the purpose of the approach section. So based on what you've read or just some of your ideas you have so far, what do you think is the purpose of this section? Why is it so important? Okay. So I promise this graphic is related. We have written our research idea and our research aims, right? So we know where we want to go. We have that bullseye. And the approach section is essentially laying out the plan for how we're going to get to that endpoint of accomplishing that research goal. So like we talked about in the overview, that's why it's so important, because you're making a case here for that what you're doing is actually feasible and you can do it. So really the approach contains enough details to convince reviewers that you can do what you're proposing. So if we think about this image here on the right, when you've given them your overall research idea, you've given your specific aims, they have a general idea of what you're trying to do, right? But they don't have any of the specifics yet of how you're actually going to do it. So that's what you're doing here in this approach section, is providing those details to help the reviewers see more clearly, okay, I know what they want to do, I know how they're gonna do it, and yes, this all sounds reasonable and it's filling a knowledge gap, so we're gonna go ahead and fund it. So that's why this approach section is so important. And it's specifically providing details that haven't been provided so far. So what you're doing really in this section is outlining the planned experiment to address the research question that you have. So remember, we have our overall research question, and then underneath that umbrella, remember that was that analogy I kept using over and over again, we have our research idea, and then we have aim 1 and aim 2. And really what we need to do then is for each of those aims, explain the approach and how we're going to accomplish those aims. So even though those two aims are related because they fall under that broader research question category, each of those aims still needs some kind of different experiment or some kind of different approach to accomplish the aim. So when you're writing your approach section, make sure you're taking into account and explaining for both aim 1 and aim 2 how you're going to accomplish those. And overall, what you're doing in this approach section is discussing how you will obtain and analyze the data you collect. So first, let's start with obtain. That might entail different things depending on what particular type of study you're proposing. Obtaining that data might involve, first of all, collecting samples from the field. These images are both from that I've taken two of my undergraduate students in the lab, Amanda on the left here and Lisbeth. And so on the left picture here, this is actually us collecting some C. funebralis snails in Bird Rock, one of those Southern California collecting sites. So for our studies, one of the first things we need to do is actually get the animals from the field. So obtaining data, that's one component of obtaining data, is physically getting the samples. And then again, depending on what type of aim you're proposing or what type of experiment you're doing, you might also from that point need to obtain the molecular samples for your specimens, right? So we've collected them on the left from the field, and the next thing we need to do is actually take a tissue sample. So that's what they're doing here in this picture on the right, is taking a small piece of the foot of the snail to use as a tissue sample that they can then extract DNA or RNA from. So when we're talking about obtaining the data we collect, that can often be several different steps, from physically collecting animals in the field, getting some kind of sample, and then extracting the molecular, either DNA or RNA or whatever we're proposing to study in this experiment. And then once you've obtained that data, you also are describing how you're going to analyze that data you collect. So often that will be something like using a certain program or an RStudio package or something like that to actually analyze that molecular data that you gathered. So in the last part of this video here, what I'll do is describe the types of information that should be included in an approach section. First of all, as we've touched on a little bit already, what you really want to do is provide an overview of the experimental design for your study. So on the right here, again, this is a flowchart from one of the red abalone microbiome projects that's currently ongoing in my lab. And even though in your proposal it's a written document, it's only 2 pages, so we don't have space to include figures, but I do actually recommend, just for your own purposes, to make sure you have a good handle on what type of experiment you should be doing. I suggest drawing out a flowchart just for yourself. I think it'll be really helpful in terms of thinking about the different steps of this experiment that you're doing. So here in this flowchart example on the right, again, this is from the red abalone microbiome project that my master's student Emily is working on. So for us, first what we need to do is collect feces from individuals. We then need to expose them to a heat stress. We then collect feces, because if we think backwards, one of our aims is comparing the microbiome of individual abalone before and after they experience heat stress to see how that heat stress might change the microbiome. So we need to make sure we're collecting feces both before and after that heat stress. Another main component of this particular experiment is comparing the microbiome of individuals that survive or die following a heat stress. So once we do that heat stress exposure, we then need to separate out the dead individuals versus the surviving individuals. For all of those individuals from both those pre and post heat stress fecal samples, we then need to extract DNA. We then need to prepare our library for metagenomic sequencing. We'll talk a little bit more about the library preparation in the next lecture, where we talk more about the logistics of genomics and transcriptomics, but this is a component of high throughput sequencing, is library preparation. And then when we get that data back, what we're going to do is look at the gut microbiome differences between dead and surviving individuals with 2 main purposes. So remember, when we're doing metagenomics type of studies, it gives us 2 main pieces of information. First of all, what type of microbes are there, so we can identify the bacteria that are there, and then we can also look at what they're doing. So because we're sequencing all of the DNA of that microbiome community, we can also identify certain metabolic pathways that they might be using. So again, you don't need to put a flowchart in your actual proposal, but it's a good way for you to work through that experimental design to make sure you understand the different components that you'll need to explain in your approach section. So this experimental design is really the overall picture, and then in the next couple slides I'll talk about some of the details you want to make sure you also include. So in your approach section, you want to make sure you're including the sample sizes and the number of sites or populations that you're surveying. This is an example from that DD-RAD study that we've worked with in lab previously. So in this case, I would want to make sure I'm explaining that I have a Northern California population and a Southern California population. For each of those, I'm sampling 3 different sites, 3 different geographic sites. And then from each of those 6 geographic sites, I'm sampling 15 individuals. So that's the example of the details you want to provide in terms of sample size and number of sites that you're focusing on. You also want to make sure you're describing what particular molecular loci you're going to be using. So again, this depends very much on what type of experiment you're proposing. If you're proposing to look at a single locus, like a heat shock protein or a mitochondrial gene, really that's totally fine. You're just going to be focusing on one particular locus, for instance, one loci. If you're using a high throughput sequencing approach, like what is shown here, then it's not necessary to say the number of loci that you're using, because really if you're doing a genomic study or a transcriptomic study, which we'll talk more about in the next lecture, you're sequencing everything, right? So it's really, you don't need to say, I anticipate we'll sequence about 16,000 genes. So the number of loci really depends on your approach that you're taking. If you're focusing on a single locus, make sure you make that explicitly clear. If you're taking a whole genome sequencing approach, for instance, you can just say that. You also want to make sure you're including the software used for data analysis. So in this, when you're writing your approach, you don't need to know necessarily how to use that software already, but what you're doing is based on literature or other studies that have done similar things to what you're trying to do. You want to use those same type of packages and data analysis for your project. So again, if we think about maybe if you're looking at genomic diversity, genetic diversity, and if your loci are in Hardy-Weinberg equilibrium, you might want to use a package like diversity or something like that. So again, because you're trying to convince the reviewer that you can do what you say you're going to do, it's important to provide specifics in terms of how you're going to analyze your data. So overall, really what we're doing here is coming up with a plan for carrying out the proposed activities in your proposal that are well reasoned and based on sound rationale. And you accomplish that by reviewing the literature. So I know all of you have already been doing that. So you probably already have some papers handy that you can reference for coming up with this approach section of your proposal. But really the way you make sure that your plan is based on a sound rationale is looking in the literature and seeing what other similar studies have done. So if you were interested in looking at insect species and whether there were certain loci or molecular evidence of local adaptation, you might use this paper as a reference where they were looking at local adaptation in bumblebees. So again, you would look through that paper, see what approaches they took, and use that to inform what you are proposing. So those are some of the basic things you want to make sure you're including in your approach section. In the next video, I'll walk us through some steps of how you might actually come up with that information.